introduce Willow Coyote-Maestas, who is a graduate student at the University of Minnesota in Daniel Schmidt's lab. Before that, he was an undergrad at Evergreen State, where he had a whole bunch of really cool research experiences, including studying how vitamins and metabolites traffic in ocean ecology and a bunch of other things. Then he came to University of Minnesota, where he has been studying at the interface of protein science and protein engineering and data science and machine learning. I met him this summer at the Proteins Gordon conference and got to know each other a little bit then. And then he got to be friendly with a student of mine, Robbie Diaz, at HHMI Gilliam Fellows meeting late last year. And then Willow reached out after that because he had some family illnesses and he has roots in the Bay Area, to see if he could come visit my lab for a little bit. And we got discussing the Hannah Gray Fellowship and other opportunities. And he came to visit the lab earlier this year and then sort of has been stuck here ever since because of the developing situation in the world. And it's been really, turning lemons into lemonade. One of the really great things about this pandemic is for us, is we've gotten to spend a lot more time getting to know Willow and having him be a part of our lab's community. And he's just a fantastically creative scientist. I will say, because he'll be too modest to say it himself, this talk probably represents like 1/10 of the data that Willow has generated during his PhD. It's just been remarkably productive and just uncovering all kinds of cool insights about the nature of how proteins evolve and how allostery functions. So with that introduction, please join me in welcoming Willow.
Can you see this? Is it working?
I can see your mouse and I can hear you.
Great. Thank you, Jamie, for this opportunity to give this talk, and in general for hosting me here at UCSF and the whole Fraser lab. And I'm really excited to be sharing with the broader community what I've been working on in my PhD, or at least part of it. And what that has been is developing and applying protein motif insertional methods where we insert motifs into all positions of a gene, for example, an ion channel, to explore and engineer its allostery and to study the general principles of protein recombination. I've been doing this work in my PhD in Daniel Schmidt's lab as one of his first grad students. It's been a great pleasure to work with him. He's been a great advisor who's allowed me a lot of freedom to go in many different directions, but also support when I needed it and to help understand all this strange data we're getting out. But first, I want to contextualize this broadly in thinking about how I think high throughput protein biology is starting to open up whole new types of experiments that are going to revolutionize different areas of protein biology. For example, this really beautiful work from Gabe Rocklin and his postdoc at Dave Baker's lab at University of Washington, where he synthesized many variants of artificial proteins and then made many mutations to measure stability and study protein and synthetic protein evolution or engineering. And then also George Church's lab has done some really interesting work, for example, in viral capsids to look at their structure and function using multi-parametric assays. But I think that going forward, we're going to see many other areas of protein biology where that somewhat necessitate, because of their esoteric nature, high throughput quantitative approaches such as allosteric regulation, where you have higher order interactions, that we need some way of perturbing the system in many different ways, or protein folding or trafficking. But what I've worked on in my PhD is to try and understand what principles are there in domain recombination and apply an empirical approach to studying this problem, as opposed to just taking observational approaches, which have been very beautiful, influential on me. I want to just understand what physical constraints are there, and why, for example, are domains mostly inserted on or recombined at the ends of proteins and not on the inside. But there's many other questions in the space. And the reason why this really matters is protein engineers, we like to recombine proteins with useful functions to make tools in therapeutics, such as taking antibodies and T cell receptors and combining them together to create chemotherapeutic CAR Ts, or GFP and calmodulin to create a calcium sensor for neuroscience. And most of this work is really focused at the ends of a protein, where you take one domain and you attach it and glue it at the end. And this has been quite useful in some instances, but unfortunately, because regulatory networks are not at the ends of a protein and are more at the inside, this doesn't always work so well. So for these reasons, protein switch and biosensor engineers tend to insert domains inside a protein to harness and take advantage of its regulation. But this is very hard because, one, it has to be able to be inserted at all, which can be quite challenging. But then two, you have to be able to know where on a protein you can even control it, which we don't understand yet. And so for this reason, most approaches have been trial and error, either low throughput, where you have a handful of regions that you know has some sort of functional relevance in the protein, and you mess around with linkers until you get something that works, or high throughput. Some really beautiful work from Dave Savage's lab at Berkeley has been to insert a switchable or a sensor domain into all positions of protein and just find something that you want. But in both of these cases, we're not getting better as we iterate through this. We aren't improving our efforts, and we don't understand why sometimes it works and why sometimes it doesn't. And for that reason, I wanted to start to try and learn the principles of protein recombination empirically, where we take many recipient proteins and we insert motifs into all positions of the protein, and then measure what that impact is on the recipient protein. And then we measure features of these motifs and recipient proteins, and we can ask the question of how do these combine to give rise to protein recombination, using, say, quantitative machine learning approaches or qualitative comparisons. But before I go any further, I want to just define insertion permissibility, because I use this term a lot throughout my talk. And what we use this to mean is the ability for a recipient protein to accept an exogenous protein motif. Primarily, the way we've been measuring this has just been surface expression, so whether a channel can make it to the surface or not because of their membrane proteins. The general way this assay works is you generate a library of variants where you insert a motif at effectively every position, and then you just measure whether it makes it to the surface or not. We measure surface expression with fluorescent labeling and cell sorting, where you use a fluorescent antibody on surface expressed channels, and then you just collect cells that are with surface expressed channels versus those that aren't, sequence to get those two populations, and then you can calculate or get the genotype or the insertion position and the type of motif to the phenotype, whether it makes it to the surface or not. And when we were starting, the ways we generated libraries was using this method from Dave Savage's lab, where you use transposons to deliver an antibiotic cassette and then cloning to replace that with your domain of interest. However, unfortunately, these libraries are not ideal. So for instance, these were 4 different genes that we inserted into and positions along the genes, where I found everywhere in red were positions that were missing insertions. So we wouldn't be able to see these in our libraries. And other positions were present 1,000 times more than others, which limits your signal to noise. And that meant in some cases, you got libraries that were effectively unusable. And this is really unfortunate. So I wanted to get around this problem and solve it, and develop libraries that are bias-free, targeted, and have full coverage. So effectively the perfect libraries for future work. And I did this using synthetic oligos that you can just print or order with whatever sequence you want. And we just designed them so that there were genetic handles inserted at every position that can then be replaced with whatever domain we want in multi-step cloning. And we call this saturated program insertional engineering, or SPINE. If you want to know more about it, it's published here, or I'm happy to talk about it at a later time. But what really matters is if it works, and it does. So these were the libraries before, and these are the libraries now. We can see there's more even coverage, and nearly all positions are contained. And the problems with the libraries before really stemmed from intrinsic bias that exists in transposons, where they have a preferential insertion sequence that's well documented. And it makes it so when you replicate library generation, you effectively get the same library. So if you're missing a position, you can't just replicate it to get that position. Whereas our method doesn't have strong bias, and also as a result, the libraries when you replicate them, they look quite different. And so if you're missing a position, you could just replicate it and fix the problem. What matters is whether this improves our data, and it does. So on the left, this was the data before. So everywhere in green here is positions that were missing data. And then orange should be permissibility, or the ability to surface traffic. And there's just this residue that looked slightly orange or slightly permissive, but it wasn't very strong, so we weren't confident in it. Whereas now we have this much stronger signal in these regions. We have more dynamic range, and we can see this whole pocket allows for insertions, which allows us to feel quite confident that we could stick something in there. So it's more complete and has higher dynamic range. And having this improved method has really allowed us to expand outward and expansively and exhaustively test insertional space. So we're now inserting 700 different motifs into our protein of interest to really get at what the principles are for protein recombination. I'm not going to go into all of these, but we just took as many possible motifs as we could think of, including commonly recombined domains, natural and synthetic domains, and small flexible peptides. And these broadly fall into these categories of small flexible motifs, well-folded domains, and transmembrane associating proteins. And the recipient proteins that we chose were CR2.1, a channel I've worked with extensively, and then 2 cytosolic proteins, because I wanted to know what the universal principles are for protein recombination and test it in multiple systems to figure out what's idiosyncratic and what isn't. These we're still waiting to collect, and David, a really great grad student I've worked closely with for the past couple of years, is starting selections in Minnesota. And then this is what I'll be discussing and what we've mainly collected. So before I show you that data, let's just try and ground ourselves in some model-based thinking. So this is a toy protein with some beta sheets here. So very structured, rigid, some flexible termini here, and say a flexible loop. We can imagine if we stick different motifs in here, that in the termini, probably everything will be allowed there. The protein will still be able to fold, and we call this class universal permissibility. Then we can imagine sticking something into the middle of the beta sheet, and we would expect all of them would fall apart. But finally, we'd expect in, say, a flexible loop that certain insertions like the square here would cause an unfolding where others would be allowed. And the reason why we think that, or why we'd expect this, is that what we're really measuring is the ability for the protein to deform in response to an insertion. So for example, in this terminus, there's a near infinite number of conformations. So you'd expect that anything can be stuck in there. Whereas in a beta sheet, you really only have one state that can be there. So you'd expect most insertions will break it. Whereas in this flexible loop, you maybe would expect that certain insertions would be allowed while others wouldn't, because there could be just a 2-state situation here where, say, an alpha helix and a flexible loop. And I did some predictive modeling, and that grounds this. I won't show you that data because it's complex and hard to understand, but these are some of the takeaways from it. 1, the recipient protein determined the motif behavior. What that means is that certain regions behaved one way and other ones behaved other ways. So in the termini, you get certain behaviors. In a beta sheet, you get others. In a flexible loop, you get another sort of behavior. And within those regions, then motif properties become important. We find that contact density and flexibility are most important at the insertion position. So that's the amount of space or the packing there that you'd expect lots of residues in a region that'll cause an unfolding or not be amenable for an insertion. And flexibility is pretty intuitive. We're measuring whether it can deform again. And finally, something that's not necessarily, I didn't think would be the case beforehand, but makes sense afterwards, is motif length seems to interact with other features to enable permissibility. And this I explained to mean that, say, motif length by itself, you can know you have a really long peptide, but that doesn't tell you very much. But if you know that that peptide has hydrophobics in it, that could allow for a hydrophobic core, which will allow stable folding and will cause one form of motif or one sort of behavior. And I'm really excited about this outcome, and I'm excited to dig more deeply into it once we have more data sets. But this is what the data looks like. It's super complex. I'm going to take time to go through it. So on the x-axis are positions of the protein where we're inserting, and on the y-axis are inserted motifs. So this is the 700 motifs and 400 positions or so. And then colored in red is whether it can make it to the surface or not, or accept a domain. And blue is whether it unfolds. And we see broadly this data breaks up into several groups. I did not decide these groups. This is all unbiased. And so these groups really are small flexible motifs where everything's allowed. These are 30 amino acids and shorter for the most part. And you see no differential sensitivity or differential permissibility in these regions. Anywhere that allows an insertion allows any insertion. There's no sensitivity. And then you have these viral filaments and transmembranes, which are proteins which are very hydrophobic and non-polar. And they have really distinct insertion profiles. Some can be in transmembrane regions. I think there's something to be learned here about how, in these idiosyncrasies, to understand how to assemble proteins from hydrophobics. But finally, there's these well-folded domains, and this is where everything that's useful is. This is where switchable proteins are, anything that's engineered, commonly recombined proteins. And these seem to behave in really unique ways. One, they have N and C termini that are strongly positive. What does that mean? That means that the domains are actually beneficial in these regions potentially. And that could follow a model presented by Jane Clark, where folding of domains can catalyze the folding of other parts of proteins, which I'm really excited about. And the other place this is useful is that we see a lot of differences in permissibility in these regions, which we could learn from to learn better how to engineer domains to be better amenable to recombination, or how do you change a domain to, in a small way, to make it behave better in certain regions and fine tune it. But then I was seeing all these interesting distinct patterns in certain regions. So I wanted to see if I could use this method to break up a structure based on its physical properties. And so colored in red, this is just all unbiased again, that I can identify regions that are disordered. So, and these are regions that allow most insertions, like the N and C termini. In blue are regions that are necessary for folding. So this is like transmembranes, these beta sheets, which are probably holding it together. And then finally in yellow are regions that can undergo, are sensitive to insertions. And these are highly enriched in regions that can undergo disorder to order transitions, which this all follows this model that I proposed before. And I think these regions, these differentially permissive regions, or regions that undergo order to disorder changes, are really interesting because that's where a lot of regulatory sites are. That's how some catalytic sites behave. A lot of the most interesting things in biology are places that have limited numbers of states. Then we wanted to test this, see if we could continue to do this in other protein systems. So I collected data sets in 3 other channels, KV 1.3, a voltage sensitive channel, an acid sensitive channel, and an ATP channel. I'm not going to talk about them, their biology. More of these are just other test cases, and we use the same assays as before. And again, we can classify their architectures based on the rigidity of the architecture. So this is really exciting to me because we can start to identify parts of a protein that are involved in folding, but it's also very early days. We've really discovered this in the last couple of weeks when we've been playing around with the data, finding new ways to mess with it. But overall, in this first part of the talk, I just wanted to give a broad sampling of different ways we can use these high throughput methods to do a lot of interesting measure interesting properties of proteins and learn something. So for example, we're learning the principles of domain recombination right now by exploring insertional space exhaustively and starting to learn why and how proteins recombine the way they do, which will be useful for engineering and for just basic biology. We're starting to classify architectures and learning we can use this as perhaps a structural biology method that could be quite useful, because right now, I don't think that there's an unbiased way to identify whether every single part of the protein, what that protein is doing, and or rather whether it can undergo conformational change. But now I'm going to focus a little bit more, focus more on the evolution of Allosteric in a specific protein family, and a family of ion channels, so that we can have a little bit more to sink our teeth into. And the ion channels ion channels overall, many of us know this, but they're membrane proteins that are integrated into the membrane that control the flow of ions down an electrical chemical gradient. They have ion-specific selectivity. Some channels conduct some things, others other ions. They have diverse regulation that all of these work together to underlie electrophysiology so that we can have complex behavior like thought, or that I can give this talk or you can listen to it. And the channel that I've mostly been working on is the inwardly rectifying potassium channel. That preferentially moves potassium into a cell to help maintain the resting membrane potential of a cell and limit excitability. When it breaks, you get things like seizures. And what I find so interesting about this family, and this is generally true about many proteins, but that there's such remarkable shared architecture across different homologs. So these are three: CUR2, CUR3, CUR6. And in this family, there's quite divergent allosteric regulation. For example, CUR2 is regulated by just PIP2, and all of them are. But CUR3 is regulated by G beta gamma. So there's GPCR activation, let's go, of G beta gamma, and you get cell signaling control of potassium current. And then CUR6 is regulated by ATP, and this is involved in diabetes. But what I find so striking about this is how can you have both shared architecture and different allosteric regulation when a protein structure and dynamics really underlies its allosteric? This is true in many protein families, but I still find this to be a little surprising. And so I want to just focus on these two, the CUR2 and CUR3, where one is a PIP2 binding, one has a PIP2 binding site, which can be seen here in these beautiful structures from Rod McKinnon's group, where you have PIP2 binding between these two helices, and then the other is at a G beta gamma, has a G beta gamma binding site right at these DE and LM loops in CUR3. But what we know about this is that CUR2 cannot bind G protein. So there's no binding interface for the G protein, which can be seen in this Western blot, where CUR2 is missing, cannot pull down G beta, whereas CUR3 can. So this one can bind, that one can't, and it's regulated. But what we know about these regions is the flexible loops have beta sheets that form an alcohol binding pocket in both of them, which is not physiologically relevant in CUR2, but it still can bind a synthetic analog or synthetic alcohol, MPD, that exerts some functional impact on it. And CUR3 has that same potential to do so. And that makes me think that potentially the DE and LM loop have some allosteric potential. And this allosteric potential could have been used to evolve G protein regulation in this protein family, because you can't have, to provide it a selectable advantage and a reason for G protein binding to have some sort of impact, you need to already have allosteric regulation. So I think this is an interesting model for thinking about the evolution of allosteric, where you could have the latent potential to be an allosteric site, and then you could evolve a site, and then have a new cell signaling phenotype that allows diversification. But generally, I wanted to ask these questions to start to explore this. Which one, does the DE loop and LM loops in CUR2 have allosteric potential at these sites? Do other regulatory sites from homologs have allosteric potential? So not just this site, but say the ATP binding site from CUR6. And is there a way we can measure this in an unbiased way so that we're not just looking at the regions we're interested in, but we can really test whether there's something special about regions that are used in allosteric versus ones that aren't. And first, let's just think about allosteric sites. By definition, an allosteric site must be able to change states in response to a stimuli. So it must be able to change states after, say, ligand binding, and that has to be propagated to another part of the protein. But if we just focus on the allosteric site, it has to be sensitive to perturbations, but it has to be able to allow some. And I think this falls really well into this category that I presented before of differential permissibility, where certain perturbations are allowed, other ones are not, and are going to be too disruptive. So there's a sensitivity that's underlined by certain places have limited states they can sample. And what's a really interesting observation is this yellow grouping, which again was sites that were differentially permissive, happened to be highly enriched in allosteric sites in this protein and homologs, which implies that perhaps we can use this to measure it. When we were starting this, we developed these assays that I mentioned before, where we generate in a library and we measure whether it can make a variance. We measure whether those channels can make it to the surface or not. And to insert domains, we just included 3 domains to start with. And one's a light switchable domain that binds to Cry2 with light. Another is PDZ, which is very well folded and been used in these sorts of insertional profiling experiments before. And this final one is a flexible linker, a GSAG linker, which I think these kind of fall into 3 distinct categories: a switchable one, a very well folded, rigid one, and a flexible one. This is what the data looked like at first, where everything in green is positions that allow for an insertion and so that the channel can make it to the surface, where everything in orange it can't. I'm just going to focus on 2 regions here. The DE loop, which as I've mentioned is the binding interface for G beta and homologs, where we see a small linker can be allowed, a PDZ domain can also be allowed, but this light switchable domain that's the same size as PDZ cannot be allowed. So there's some sensitivity here. And then the other site it allows is these beta sheets where here and here you can insert CYP81. You can't insert PDZ quite as well, but you can a little bit. But you can insert a small flexible linker. And that's I found unintuitive and surprising, but it implies there's a sensitivity there. And that site's a ATP binding site in homolog. And the other homolog that I showed, we look at this on the structure, we can see that there's a lot of differences between PDZ and CYP81, where you get where you just get different signals here, which is surprising to me initially because these were the same size. I thought the same amount of space it'll be taking up in three-dimensional space, more or less, should get the same profile. And so we wanted to calculate the difference between that to get at this differential permissibility I mentioned a couple of times. And that's we just calculate at every residue the difference between them. And we use a Euclidean distance, or effectively an absolute value like score, where higher means more different, lower means less different. And then we can break up the protein into functional residues, or ones involved in regulation, and scaffold residues. So for example, a functional residue would be this DE loop, where we can see there isn't a strong signal there, and there is over here, so that's very different. And then another would be the scaffold transmembranes, where you can see there's about the same signal in both of them. And when we look at these sites across all inward rectifiers, allosteric sites and ones involved in function are more differentially permissive. But we really wanted to, once we had improved our methods in ways I mentioned a little bit earlier, we wanted to we wanted to get cleaner data and then really test this hypothesis further, because before our data was a little messy. And we did this, and we've been calculating bulk properties now across many more motifs. We're using 15 motifs or domains, and we can calculate mean or overall permissibility and variance or differential permissibility. If we look at differential permissibility first, we can see there are two regions that have strong differential permissibility, and mapped onto the structure, they're here and here. What's exciting about those regions is those are PIP2 and G protein binding sites that I've mentioned before. So this is in support of the DE loop having some latent allosteric potential, or it's sensitive to a type of insertion. Then working with the collaborators, Vincenzo and Antonio, MD simulation experts, we did simulations of the APO and PIP2 bound states of CUR 2.1, and then calculated the change in motion between those two simulations and mapped that onto the structure. And what we find is that the places that have, there's two types of, these regions have large changes in motions that are correlated with PIP2 binding, or in other words, activation of the channel. And this implies that there is some sort of coupling or motional coupling, and this is in support of the D loop having some latent allosteric potential in these regions. And then if we look at the mean permissibility, we can see that there's clearly this path of residues that connect these two regions together, which perhaps implies a mechanistic or explanation for why this insertion here can be propagated to another part of the protein, and that there is some sort of allosteric coupling here. And then we looked broadly at differential permissibility and started looking at where are those sites coinciding. And we can see across this, differential permissibility map that sites that are used in allostery, both in our protein and close homologs, appear to be differentially permissive. So it appears, which implies that perhaps that there's latent pools of allostery that could have been used in evolution to diversify this ion channel family. And furthermore, that we can use this method to potentially measure the allosteric potential of architectures of proteins. But we really wanted to test whether these sites are allosteric, and we already included a switchable domain. So we inserted our switchable domain into sites with predicted allosteric potential. The places where we expected to see results or predicted were going to be this DE loop here, which is, I had mentioned already, it's the binding interface, and CUR3 for G beta. And then there's alpha helix here that's potentially used in regulation by G alpha. And we had inserted the CYP81 in there, and we wanted to test whether light recruitment of Cry2 would modulate function. So this is very similar to how G beta gamma recruitment activates this channel potentially in the homolog. And I wanted to measure this in high throughput, so I wasn't just measuring a single variant at a time. And to do this, I developed an assay that's based on if CUR 2.1 is present, it'll shift the membrane to be more negative than if it's not. and I can read out the shift using a fluorescent voltage sensitive dye that partitions into the membrane and becomes more fluorescent, and then use flow cytometry to see this change in fluorescence. So for example, no Kerr 2.1, present Kerr 2.1. I want to highlight that in comparison to patch clamp electrophysiology, which if you're very good at it, you can get through say 20 variants in a day, or sorry, 20 cells in a day. In this case, I can measure 10,000 cells in a second. So there's a massive gain in scalability here, though of course there's a little bit lower resolution. But really the question was whether there's allosteric sites here in Kerr 2.1 and whether we can control them. So we tested this. So function, the assay that I mentioned before, is on the x-axis. So this is a wild type, for example, and a dead mutant. And then on the y-axis is whether it can switch with light or not. And we can see there's 3 sites that's different, that significantly can switch. One is this 186 mutant, which is within the PIP2 binding site of this protein. So it's a known exploited allosteric site of this protein. Another is this 362 mutant, which is right in an alpha helix that's used potentially in a homolog for G-alpha binding, but it's only been seen in NMR structures, so it's a little controversial. And then there's this 238 mutant, which is right in the D loop. It's the most exciting from a biological standpoint, where it looks like there is some intrinsic potential, allosteric potential here, though we need to, this won't necessarily be useful as a tool, but we wanted to validate whether these two switch, these more significant switchers could switch with patch clamp. And we can see that as a baseline, you see some openings here. So these are channel openings, and then you shine a light, they go away, and over time it recovers back. So this is an activation with light. And then over here, we could see, down here, we could see that there's activity. So opening and closings, opening and closings. You shine light, it drops down quite dramatically, and with time it can recover back down. And you can see this really beautiful histogram in this case. This one here, I think, could be a very useful tool because these channels help maintain the resting membrane potential and set them. And we could imagine using light to set as if it's a potentiometer, the exact resting membrane potential of a cell. It would take some optimization, but I think that there's potential here that I haven't been working on because I've been more focused on the insertional profiling side of my project. But in summary, we've been developing these insertional profiling methods, which really allow us to learn something about the exploited and latent allostery of a protein, which combined with protein dynamics, we can start to really hypothesize that there's some coupling here that may have been used in the diversification of regulation. And we're excited to continue to dig into this and validate this with other methods and test this on other protein systems, of course. And finally, we're able to engineer allostery with this method and generate tools. So overall, these pipelines that I've developed in my PhD could be broadly useful for basic biology to understand allostery, evolution, could be applied to engineer protein switches and sensors. And then finally, we could perhaps imagine that if we can identify allosteric sites, that we could start to use this as a coarse grain method for identifying potential druggable sites or for drug development. But in the future, I really want to study work and continue to work in this high throughput protein area that I think there's these areas that I mentioned at the beginning where a lot of these problems these areas have been challenged based on the lack of high throughput quantitative approaches to study these very complex problems. And I think that these areas and many others are going to be somewhat revolutionized by high throughput approaches. And I will probably be using more deep mutational scanning, which some of you may be familiar with, where you can make every single possible single point mutation of a library and then measure the fitness of it. And that's been quite useful to identify whether mutations can break a protein. For example, by measuring protein abundance, which was done here using a split GFP system that allowed Kenny and his postdoc to identify pathogenic mutations. But I really want to push the field forward by building a technology platform where we can not just measure if a mutation affects protein function, but how it does. And with that, by developing multi-parametric deep mutational landscapes where we can understand broadly how mutations affect function, and then develop mechanistic models of disease and biology to understand these proteins better and be able to fix them, of course. And this is important in proteins such as TRPV1, which can break in many different ways. For example, a mutation can impact surface expression, but not total abundance. So abundance only a metric would not work in this case. And of course, mutations can tune a protein's function quite dramatically by changing its kinetics or its activation by different regulators. And so channels are a great family to think about. How can we measure the multi-parametric aspects of its function? And an example application of this that I wrote this up for Hannah Gray, application that I'm a finalist for now. It was to try and explore a question that's been a long-held question for David Julius here at UCSF, of course, and Yifan, trying to understand the structure of it as well, which is to understand how does TRPV1 respond to temperature atomistically. And we could think about using these deep mutational landscapes to find variants that have altered properties and then validate them and solve their structures to really get at this bottleneck I see in structural biology, which is finding variants that are trapped in different states, especially for TRPV1. And using all this together, we could start to try and develop a integrated model of temperature activation. And when I was writing this application, I was thinking about it in terms of a postdoc where I'd learn new skills. So I wanted to learn cryo-EM and learn patch clamp. But what I realized, as I've mentioned before, is that really these high throughput approaches are going to revolutionize parts of protein biology, and I want to be a part of that. So I'm going to focus on this area, and then these areas, and then I can happily collaborate out and work together with other members of the UCSF and other communities to try and work on this and many other problems like it, of trying to understand complex phenotypes and develop these high throughput assays for those purposes. And something that I started to work on before the world shut down was to try and measure TRPV1 activation with a fluorescent reporter. So there's these fluorescent calcium sensors, such as GCamp, where I took 2 mutants I got from a friend who's at the NIH and soon is going to start his lab at UT Austin, where Andreas, who that one was a temperature shifted sensitivity mutant and one's just wild type. And in here, we can already, with this fluorescent sensor, detect the difference between them and potentially detect a significant but not sortable difference on temperature sensitivity. And I'm confident that with a more optimized system, I will be able to start to detect, be able to use this to sort on these different parameters, and we can really develop a multi-parametric functional landscape. The other area that I've mentioned a couple of times is trafficking. TRP channel expression can really be disrupted at many different steps. It's a long way from it to get from the nucleus to get to the cell surface. There's many steps along that. In many places it can be broken and altered. So for that reason, I'm going to develop a platform to measure trafficking using a split fluorescent protein, where I can take one half of that and attach it to a localization signal peptide, and take the other half of that fluorescent protein, attach it to my channel to create a trafficking biosensor. And what's nice about this is we could then, in parallel, be able to quantitatively measure how much of that channel ends up in every single part of the cell. And then from that, get a landscape to understand how do mutations alter this landscape overall. And this method, of course, isn't specific to TRPV1. It could work on any protein, and I plan to expand out to other proteins with interesting trafficking defects like CFTR and many others. And we can also, we will combine these with genetic and chemical perturbations to study the complex network of interacting proteins that leads to the trafficking landscapes we see today. And I think this will really open up a lot of space in biology to quantitatively understand how proteins end up where they do. And more broadly, I want to develop this pipeline and many other pipelines to study how, not just how proteins, if proteins break, but how they break, to improve our understanding of biology and improve therapeutic approaches. With that, I would really like to acknowledge the people who contributed to this work and have helped me along the way, and you, my audience. Thank you for listening. Especially I'd like to acknowledge Daniel, my advisor, who's been really great and supportive and allowed me to move out here too, and been supportive of me coming out here and spending time with my family and getting this opportunity to be in the Fraser lab that's blossomed out to me giving this talk, and hopefully many other things. Young Wee, our lab manager, who's built the foundation on which all of this exciting and many other projects have been built on. He's great. David, extraordinary grad student who I've loved working with, and working closely with, and it's been such a pleasure. Stefan, an undergraduate that I trained, and it's been so wonderful to see him go from pipetting to learning to pipette to now playing a role in projects. Jamie and the rest of the Fraser lab for helping to welcoming me here to California and UCSF, and giving me these amazing opportunities and giving me great feedback on my project. People in the flow cytometry core who taught me how to do the sorting and who've become very close friends of mine, and it's been so great to work with them. My co-advisor, Chad Myers, who's an expert in big data and data mining and how to deal with these large data sets. Without him, I would have been completely lost because I didn't know how to code when I started my PhD. Vincenzo and Antonio, MD simulation experts who really helped us. Doug and Kenny, who gave us reagents before they were published so that we could push our project forward, and my funding. And with that, I'd be really happy to answer any questions that's built up.
Great. So thank you so much, Willow, for a great talk. I'm sure people are applauding in their own rooms right now. Give you a second to get a drink of water, and then I see some stuff coming through, and I'm going to enable my colleague John Gross to ask his, hopefully enable. Oh, I don't seem to have the controls that I had previously. I don't think I was made a co-host, so I will just read his question. Yeah, so because I'm not a co-host, I can't unmute people. So I'll read John's question first, which is the reason that insertional permissibility tracks with regulatory dynamics due to increased conformational degrees of freedom that are more accommodating of diverse sequences, or something else?
I'm not positive right now because one, I don't know the answer to that 100% because we've really done this in this channel and then several other channels, and I'm waiting on that other data on those other data sets. But I think that it's due to, as I mentioned before, where is that slide? I think it's because places that are regulatory sites happen to also have some states that they can occupy, which is kind of what you were implying there, but other states they cannot. So there's this limited conformational states that they can occupy because they have to be able to respond to stimuli. I don't know if that helped answer that question. No.
Okay, next I'm going to let the person who just unmuted, who's now allowed, C. Kugantakis, to ask their question.
Hey, thanks, Willow, for that great talk. Do you think that that your results suggest that a mechanism for local unfolding and regions that have a propensity to undergo partial unfolding in allosteric mechanisms? Because this kind of reminded me of some of Vincelder's work on adenylate kinase, where they viewed local unfolding as a really important determinant of allosteric mechanisms. So do you think that techniques like hydrogen exchange could be used for protein design purposes to identify latent allosteric sites?
Yes, I do. I think that I think it would be lower throughput than something like this, but it could be very useful in like validating that these sites can undergo those conformational change. We're fortunate in this context that we have this channel where there's two different states solved, and that so far pretty much all these sites undergo some sort of change in response to stimulus. So it's easy for us, but yeah. But I that's a great question and a great line of thinking, and yeah.
Okay, next, Dan, I've given you the power to unmute.
Okay, do you hear me?
Yeah.
Yes, but speak up.
Yeah, interesting data. So I was wondering, so the first screen with Q 2.1 looks at folding, not function. And I was wondering if you have an idea of what's the proportion of each class, one versus the other. And the other thing was, just to remind you, you probably know that there's a very nice yeast transport assay for rescuing potassium transport deficient yeast, and Q 2.1 is probably the best channel that's ever worked in that assay. And it would be interesting, it might be interesting to use your libraries through that. It's also very straightforward. You can look at, you know, tens of thousands of mutants. But there you get, out the other end, you get guys that have to be functional.
Yes, absolutely. I would love to do that. That yeast screen, we thought about doing that at some point. One of the reasons we were concerned about doing that, I think that same assay, I don't know if you're familiar with Anna Moroni's work, where she's engineered a switchable channel link. But I know that channel ran into some issues with trafficking when it was first developed in a yeast-based system and then tried to bring over to mammalian systems. So when we were starting from an engineering standpoint,
But that was with KCV, that wasn't with CARE.
Yes,
you know, I did it with CARE 2.1, and basically lots of things work, right? It's really, it's very portable, because KCV has some weird issues, right? Being a viral K channel and things, but but you know, because it's important, right? Because what you really want, because you're inferring a lot about function that you actually don't measure in all these experiments, and being sort of highly regulated proteins, multi-subunit proteins, right? The issues with the channels is, in the end, like, how much have you perturbed the function?
Yes, absolutely. I agree. And I think that this is, so I've done a little bit of trying to measure function. So yeah, absolutely. I would love to do it in the yeast assay. I prefer, if possible, to just be measuring function. And I've taken that assay that I mentioned before, that was a voltage assay, and tried to develop it as a high throughput assay, but it runs into issues that there's a lot of dynamics involved just when you put a voltage sensitive dye, that it starts killing cells and things like that messes with sorting. But I have been able to kind of run the same assay again, but then collect functional channels versus non-functional cells based on the resting membrane potential, and then calculate differences between them, and or look at enrichment in different populations. And we can see that we do get function out of this. And from this assay, we also are able to see kind of the same sites coming out as being differentially permissive as well.
So how many of those have actually been looked at by electrophysiology?
We haven't looked at any of these. So this is very—
I really know, right? Because again, it's kind of a proxy assay, and
yes, absolutely. I agree that more work has to be done in that space of moving into functional assays. Thanks for your questions.
I'll take the power that I've always wanted to have in real life and mute Dan and unmute Cole.
Am I unmuted?
You are.
Cool. Yeah. So yeah, great work. I found that really interesting. I was just wondering, so this could have like two angles. Like, one is sort of biotechnology, the other actually is still like evolutionary based science. But I'm wondering if there's any sort of, I use the word confluence, but if there's any sort of similarity between sites that are have a high insertional permissibility and sites that are drug binding sites. Like, I'm just wondering if you, in a broad way, if you've checked for that kind of correlation.
I haven't checked for that. That's the simple answer.
Okay.
Yeah, I would like to. I briefly mentioned that that's something I'm really interested in trying to apply these methods for, especially in other proteins that are very desirable drug targets. Of trying to apply this method and then potentially seeing whether from that we could identify cryptic allosteric sites or something like that, because I think that there would be potential there to do that. But it's very early days. We just haven't had enough time to profile all the proteins we'd like to and get through all the data we've even generated. But yeah, it's a great line of thinking.
Okay, Robbie.
Hey, Willow, this is actually Joey. Yeah, I guess you spoke to it a little bit answering these questions, but I was just curious how you thought that your domain insertion data would correlate to the DMS data you would expect to see.
Yeah, so I think that that's an experiment I really want to do. I want to do a DMS. That's one of the last experiments that I'm going to somehow get to do before I graduate, is to try and compare a DMS to an insertional scan like this. I would expect that a DMS wouldn't quite as much detect like a difference in conformational state or something like that, because the size of perturbation of a single amino acid is going to be so much smaller than the size of like a 10 kilodalton or greater domain. Yeah, so Intuitively, I would expect very different results. And I think a DMS will also behave a lot better in like a functional assay or something like that, because it's not going to be like completely unfolding a protein or something like that. Yeah.
Cool.
Good question.
Okay, well, I don't see any other questions currently in the queue. I'll just vamp for a second to give folks a chance to chime in or indicate in the chat that they'd like to. If not, thank you so much, Willow. You know, every time I've seen you talk, I'm always so impressed at the new technologies you develop and the cool insights that you get out of them. So this was no exception. So thank you very much for talking to us today. And thank you everybody in the audience for joining us.